biofilm, because they are not normal cells, right, somewhat defective cells, and if they are added to food, are you going to get sick? So we asked that question. So we try to study the differences in the pathogenesis between sessile and planktonic cells. So as I said, I'm going to use Listeria as an example, as a model to study that. Okay. The reason we study Listeria, if you are not familiar with this organism, this is a serious foodborne pathogen, and it's not a typical foodborne pathogen, but it causes systemic disease, and you can find them in the environment, animals and humans, and they grow at wide range of temperature. They are resistant to acids and salts, a number of sanitizers or antimicrobials, and they also form biofilm, okay, in number of different environments. So we wanted to study if this organism going to have a differences in their pathogenicity. So I showed you before a little bit that if you add them tunicamycin, you can disrupt their biofilm capacity, but they can naturally form nice biofilm when you grow them in the laboratory condition. Just a little bit about its pathogenesis, means how does Listeria make you sick. And so the way to study is that it's a foodborne pathogen. And this is a typical Listeria monocytogenes electron microscopy photograph. And since it's a foodborne pathogen, you have to consume it. It's like 100% foodborne. Sometimes other form it can get into your body. And so once it is in the food, you eat it, and then it absorbs through the intestinal tract. And if you are familiar with the anatomy of the human body, so you know that we have lymph nodes called mesenteric lymph nodes in the gut, and that's where the most of the pathogens are absorbed. Then it can get to the blood circulation. You can see them in the liver, spleen, and essentially it can reach to the brain. And also in pregnant women, it causes abortion and fetal death, or premature birth. So because fetus doesn't have immunity, and that's why they are very susceptible. And this disease is very similar to Toxoplasma gondii, which is another parasite that also causes similar type of disease. Okay, and so it doesn't cause much gastroenteritis, but very little. That means you may not see diarrhea, okay, or stomach problem, but it becomes more systemic disease. The other bad news about this organism is that if you had Listeria today, you will not know until like 2 weeks or 3 weeks later, and it's a long term. And in pregnant women, it's even longer, right? Unless somebody is pregnant, had Listeria, and then next thing you know, the fetus is aborted, or the fetus is dead, or if you have a full-term pregnancy and the birth could be infected fetus, there's a question that whether fetus is going to survive. So it's a huge concern for pregnant women, and when there is outbreak, it's huge issue. And I don't know about India; they don't do some kind of surveillance we have in US, but I can guarantee you that this organism is everywhere. Okay, a lot of times the doctors don't know what caused it, what is the reason for somebody lost their fetus or premature birth, right? And a lot of those cases are happening, and I heard those stories. But the bottom line is that this organism causes systemic disease, and abortion and neonatal listeriosis is also very common in pregnant mothers, because our immune system is weak and this organism can infect. So that's the background about this organism as infection. The cattle also get infected, the sheep also get infected, and these animals can also be the source of Listeria. Okay. Now, I got to give you a little bit about its mechanism, how it causes disease. And if you look at this, it produces, if this is the Listeria producing a lot of different types of virulence factors, pathogenic factors, and all of these factors help the Listeria to get inside the cell and then get to brain or other tissues. Okay, those who don't have much microbiology background try to understand the story. Like if you have pathogens, they are getting into the cell, and they are using a lot of these virulence factors. So this is, for example, one of the important pathogenic factor and the receptor. And the other one we've been studying called heat shock protein 60, and this is called Listeria adhesion protein. So So, I mean, Listeria has to have a lot of proteins, right, on their surface. And as I mentioned yesterday, a lot of those proteins serve as a biofilm forming protein, okay, and also some of the proteins help infect host, okay. So, they have a dual function. So, this protein has a dual function, Listeria adhesion protein, and they do all kinds of stuff, so helping the bacteria to go through. So, if you take in the laboratory, this is what our laboratory pictures. All this data I am going to show, this is from our laboratory, and this is Listeria electron microscopy. So, if you grow them with the intestinal cells, because that's where they go through, and if you grow them, this is called differentiated ECO2 cells. We grow them, and then when LM touches, and you can see it's trying to get inside. His whole job is to get inside the cell, okay, because it's an intracellular pathogen. And then you can see this is already got inside. These are simple studies you can do, but this is how they get in. But more complex picture I am trying to present to you is that, imagine the bacteria is inside the lumen in your gut, right? How do they pass the gut wall? Okay, we got epithelial cells, you got mucus layer, right? And then the bacteria has to pass through the mucus and then get to the epithelial cells, right? And then go through that and get into the blood circulation, because they need to get to liver, they need to go to spleen, they need to get to brain, they need to go to also placenta in pregnant women. So, all of those places they need to go. This is their job. This is how they cause our disease. So, to make a simple statement is that how do they go through the gut barrier? That's an important question, right? Because the gut barrier is very solid. We have epithelial cell lining, and unless you cause damage, it cannot go through. And Listeria doesn't cause diarrhea, I mentioned that. It's not Salmonella, it's not E. coli. It has a totally different function. So, one of the mechanisms, we said, well, that's what we want to explore. And this looks very complex, but imagine this is one of the epithelial cells, this is another epithelial cell. So, when the bacteria comes in, we found out it binds to one receptor, just to understand the story, called heat shock protein 60. And then it initiates a series of cascade of signaling event, or it called okay, so NF kappa B activation, or it's called signaling cascade. So when the signaling cascade happens, ultimately you get some inflammatory cytokines released, and then other thing it does also, it activates called myosin light chain kinase, and it activates an enzyme. That enzyme, what it does actually moves the protein which is holding two cells together. Okay, and if you look at the cells, I will use an example. Think about the cloth we are wearing, right? These are all stitched, right, on a thread. Sometimes if you remove that thread, you can remove that, right? You pull one end and pull it. What will happen? Two pieces opens up, right? Imagine that happens in the cells. These two cells were, short like this. Okay, this is called claudin, occludin, E-cadherin, all of these proteins holding this cell line together. Okay, so it's like a closed door, right? And how do you open the door? That's the question. Because if you don't open the door, you cannot get in, right? It's pretty simple. Okay, so the way the bacteria does that is that as soon as it goes through this and it activates, it kind of sends a signal, and then these proteins, which are like a thread or like a lock in the door, it gradually opens up. It opens up because bacteria initiated that process. Okay, it's called induction, and that leads to the opening, and then the bacteria can easily go through, right? So this opening of the tight junction barrier is an extremely important pathway for many pathogens to enter our system. So if you take Helicobacter pylori, I think somebody mentioned about that. Helicobacter pylori cause gastric cancer, and also they go through pathway same way. Salmonella can also use that, E. coli also, Campylobacter, many pathogens, they have learned a trick how to go through our system. So we figure out that, and so there are a lot of proteins, these are many years of research, and then the bacteria easily pass through. Okay, this is the way to do it. Remember this mechanism, because last day or day before last, I will talk a little bit more about this, and that's the reason I'm spending time. So this is like a simple schematic showing you how, if this epithelial layer and how pathogens any enteric pathogens which pass through our gut barrier, and they use all of these pathways. So this is, we call it paracellular translocation. It opens up, it goes through it. Okay. There is another way, it's called internally mediated. This is intracellular pathway. And other thing I didn't tell you, that if you look at the epithelial cells in the gut, the cells continue to die. Okay, you know that our cells are not permanent, right? How long their life? Anybody knows? How long those cells live in our body? What is the actual lifespan, or how long you think they are surviving in our body? Anybody? What is the average life of cells in our body? So all those epithelial cells, endothelial cells, all kinds of cells we have, right, in our body, we are made of cells. What is the average life of those cells? Come on. Don't feel bad. Just give me a number. How many days, or how many years, or how many months? I mean, average life we live, 70, 80, whatever, maybe. That's not the life of the cells, right? Come on, give me a number. Huh? Say that again. 2 weeks. Okay, that's good number. Anybody else? That's very good. Did you Google it? Okay, that's exactly, yeah, about 12 to 14 days. You know, so she got the right number. So average life of the cells, this is, you should know. If you are studying microbiology or any biology, you should know average life of the cells. Because our cells in the body, they are born and they die. Okay, they are not permanent, except the nerve cells. Nerve cells have a long life. If you have a damage in the nerve, it takes forever to repair. That's why the paralysis patients, they suffer longer. But any other diseases, if you are not cancer cell, if you are normal healthy cell, you have a short duration of life. So that means our stem cells in the body continuously making these cells, right? And then these cells mature, then they die. In the gut, gastrointestinal tract, throughout the GI tract from mouth to all the way to the rectum, the epithelial cells lining, okay, they are born, they die within 12 to 14 days. Why it is important? Because in order to understand any of those pathogenesis and the microbial interaction, this is a very important information. Okay, keep that in mind. One of the pictures right behind me telling you that when these cells die and they are pushed, they leave. Okay, so it's called cell extrusion, and this is continuously happening. So body, what it does also sometimes, if pathogen attaching their cells, they will try to remove that cell from the body. So it's called sloughing. It slough off. The bacteria and the cell goes. So when you are suffering from diarrhea or say dysentery, any of those gastric problems, these epithelial cell lining totally wipes out. They die pretty quickly. Those microbes kill that. But in natural process, these cells, I think it looks like this, like a tube, right? Like a mini heel. You know, this is called villi. And those epithelial cells, they are generated in the stem cell below, they'll move up as they mature. And as they die, they fall off from the tip. And what happens when these cells die, they open up those junction in between, right? And some bacteria, if it is hanging there, they are waiting for that opportunity. So they will go through. So this is what happens. If the cell die, get out, and then Listeria can get in. Don't blame it to Listeria only. A lot of those pathogens, they get through. I'm trying to tell you how they get in, right? This is also another way. We have mucus secreted, and when the mucus is excreted, it creates a hole, right? This big hole, and then the bacteria can go through this also. These are natural process. And then the other one, you heard about M cell, microfold cell in the gut, and these cells also naturally phagocytic. Their job is to bacteria, some food also can go through those M cells to cross the barrier. Okay. Once they are underneath, they are pretty much free. Now they will encounter immune cells. Immune cells will try to kill them. So I explain you to entire pathogenic model how pathogens enter our body before they make us sick. So this is one way the bacteria can get in. There is another one also called dendritic cell. They also sample bacteria in the lumen and they bring them in. So just to tell you, this is how bacteria or pathogens, when they enter in the gut, and this is how they get into the blood circulation. That give you some idea? Big picture? Okay. All right. So what I was trying to teach you, I know these are some of these models. So we ask that question: is that sessile versus planktonic cell, are they going to have differences in pathogenesis? This is just showing the biofilm formation. The way we assay it, I think you are going to learn today about the crystal violet staining. I think that's the laboratory training you are going to have. And so if you stain these cells on the plastic or glass slide, and with the crystal violet, if they are forming biofilm, you will see this thick layer. Okay, and if you treat it with the alcohol, this dye will come out, and so you can measure the absorbance of the dye. And then if you have a more or higher concentration of dye, that means you have a stronger biofilm. Okay, if you have lower amount of dye, that means weaker biofilm. If you have no, that means no biofilm formation. So this is how you can quantify any organisms they are making biofilm, right? And so let's say I had how many? There are over 100 Listeria isolates. Then we wanted to see how they form the biofilm, if there is a difference in the biofilm forming capacity. I think yesterday you saw some data. So they vary, right? So this strain at the bottom, they are very weak biofilm former. They don't form biofilm much. But the one on the top here, it forms a strong biofilm. Same way, these clinical isolates, you can see a wide range of biofilm formation. So to study this, we wanted to see if you take a strong biofilm former versus the weak biofilm former. I'm not going through the details of all this data, but the purpose of understanding the planktonic versus sessile cells, we picked up a couple of organisms from, say, high biofilm former to the intermediate. These are some of those we picked it up to use in our study. You can compare their biofilm formation, and so then we looked at the morphology though. So surprisingly, Listeria from the biofilm, at least this 24-hour, 48-hour biofilm, we didn't see much differences in the morphology. That's kind of good or bad, but this is what we see. Not all organisms going to change their morphology. Okay, and so that's what we saw. Planktonic or sessile, they maintain pretty much similar shape and size. So one thing we wanted to see, if you have a planktonic cell, right, fresh, and then sessile cell, means we formed the biofilm, and then we scraped off. We didn't allow them to grow. We didn't give them additional nutrients. We just took the cells directly from the biofilm. You can collect them, okay, by scraping, and different way you can remove them. And you take those cells. Make sure sessile cells never convert into planktonic, okay? Then this experiment is going to fail. So you have to be extremely careful. Now you take those cells. Let's see, first step in infection is the attachment, right? They need to attach to the epithelial cell. So we use this called KECO2 cell. I mentioned that before. That's intestinal epithelial cell. And let's look at this biofilm and planktonic. If you look at the adhesion of all of these, can you clearly see the difference? This is planktonic. This is biofilm. Means the sessile cell. So sessile cell, already I mentioned, they are really different types of cells. They are not producing all those adhesion proteins. Look at this. All this 4 stain. This is one of the deadliest pathogens. It's highly infective. This F4244, and you can see this planktonic has a higher adhesion compared to the biofilm forming or sessile cells. So clearly this says they don't attach very well. So instantly told me, oh, looks like these sessile cells are not infective, right? This is what tells me. Well, let's look at it in invasion, because they need to get inside. And same data, look at this. High planktonic cells, highly invasive. Look at the percentage of cells got in. And whereas the biofilm, very little invasion. So we are totally surprised seeing this data. We didn't expect that, but that's what was the hypothesis, that sessile cells are going to be less infective because they are already under stress conditions. They are not expressing all the virulence factors. So that's what we expected. And then another thing, we said that translocation or paracellular route, this is called transwell assay. So you can grow your epithelial cells in a membrane, transwell, and if you add your bacteria on the top, if they are going through the paracellular route in between cells, and you can count them at the bottom layer, and we also looked at that, and you can again see the same result, that planktonic cells has a high level of translocation, whereas the biofilm or sessile cells are very less. So as soon as anybody sees this data saying that, oh, sessile cells are non-pathogenic or not infective as planktonic cells, we are discussing, right, that proved that point. But then the whole idea started thinking, we still get infected, right? Because these cells are pathogens only for certain moment of time in biofilm under stress conditions. They are not expressing those pathogenic factors. That makes sense, right? So we need to prove that. If I stop my experiment right here and you try to publish it, what would be your conclusion? You would say sessile cells are non-pathogenic or avirulent, right? Case closed. And then you said, well, I want to publish my paper. And people are going to believe that. Yes, your experiment was good, but that's untrue, right? In bigger picture, in reality. Because we still get sick. They still can cause disease. So we didn't want to stop that, right? So next we wanted to see, well, let's look at that in actual animal system. Let's see if this case is true. So in order to do that, this is just to show you all those protein expression I mentioned. These proteins are also not expressed in sessile cells. One of them, let's take it, virulence factor, hemolysin. They make hemolysin, okay? And if you look at the planktonic cell, makes a large amount of this hemolysin, and if you look at there in the biofilm, look at this, they didn't make barely any toxin, okay? This is more like a protein level verification. And we talked about this lab protein or internalin A protein. Also in planktonic, you will see high level. They are important virulence factors, right? And biofilm or sessile cells, they barely express. So yesterday or day before, I keep on saying that if microbes coming from biofilm or stressful environment or even saprophyte, right? You mentioned that saprophytic lifestyle from soil or some other unusual environment, they don't need those virulence factors. So that's why they turn it off. They don't make it, right? But as soon as they're in the body, they are going to wake up and they're going to make those to make you sick, okay? And so that's what happens outside. Okay, I will not spend more time. Let's do that mouse study, okay? What we did, let's design. So we have again the biofilm cells or the sessile cells and planktonic cells. You feed them to the animals orally because you get the oral infection, right? Then every 12 hours, this is 12 hours, 24 hours, 48 hours, we sacrifice the animals and look for this bacteria in liver, in spleen and other tissues to see how much they invaded. Make sense? This is how we know how well they are infected. So at 12 hours, I know this is a big table, trying to make you understand a little bit. So this is a intestinal, jejunum, ileum, cecum, colon. Then this one is mesenteric lymph node, liver, spleen and kidney. So what we are looking at, how many bacteria arrive to these organs, right? And since the number is so low after 12 hours of infection, so we wanted to say yes or no. Means we use some assays to say if the Listeria present in these organs or not. So if you look at it, the biofilm almost, look at this, 0 out of 5 mice, we could not detect any Listeria in these organs, except 1 out of 5 in cecum and colon for these, right? But planktonic, you can see higher number. You see this one? Because planktonic are active, they can spread very quickly within 12 hours. So up to 12 hours, biofilm cells, it looks like they are not doing anything, right? The cell cells. And this is also these other strains we verified, exactly similar data. But then we are asking, that's only 12 hours. Let's look at 24 hours, okay? 24 hours, we still saw some difference, okay, but these differences are getting smaller between sessile and planktonic, okay. You can still see this if you follow these graphs, okay. But the most important one, when he looked after 48 hours of infection, the story changed. Do you see this one? All sessile cells become like now planktonic, right? They are in a favorable condition. They converted, they no longer behaving like a non-pathogen. You can clearly see that they are all becoming infective. And most importantly, what happened is that after 48 hours, those sessile cells become more aggressive, more pathogenic than the planktonic cells. Do you believe that? This is kind of like a, they were sleeping giant, right? They are not infective at the early stage. But as soon as they arrived in the gut, this is gut environment. I think you are doing this, different parts of the gut study. This is in vitro simulated fluid, and we wanted to look at the protein expression and also their behavior. So the storyline is that those organisms started to express, the sessile cells started to become planktonic, and they started to express even at higher level of those proteins when they're in the gut, because we analyzed the gut sample. We took the gut sample from the animals. These are actually coming from the mouse gut, and we are looking for those virulence factor. And if you look at it, LAP and internalin A, especially LAP expression, significantly increased in the sessile cells than the planktonic cells. So what is the storyline? I didn't want to go details of this. What is the storyline? What did you learn? Sessile cells are still pathogenic, right? We all agree. And what they did, because they were in the stress conditions, remember the word stress, right? Whenever some organisms are under stress, they initially look like they are innocent or non-pathogenic or virulent. But eventually they had aggressive behavior than the planktonic. So a lot of this stress make these bacteria well adjusted or well adapted for the gut, because the gut is a stressful environment, right? We talked about acid, we talked about bile, we talked about antimicrobial agents. We got a lot of barriers in the gut, and that's unfavorable, oxygen deficient. I used that word before. And because those stressed organisms, they already adapted themselves, and when they got into the gut, they become more aggressive. And so everything we learn from this actually showing that these organisms are more virulent. So introducing stress to microorganisms, you have to be extremely careful. Try not to introduce stress to these microorganisms, because they're going to come back and going to be more virulent and invasive. And that's the importance of stress. Everybody clear? Any questions on this? I didn't want to go more details. I just want to give you the big picture. If you want to learn more, you can ask me questions later or now. And this, I thought that was kind of fascinating. It was published in Nature Biofilm Microbiome, I think some article. I don't know whether I have cited this or not. Yeah, that one. This is where this whole thing was published. Anyway, so I thought that's kind of fascinating, talking about biofilm cells. They're equally pathogenic. Okay. I think I will end it here. I think that was the last part of the. See if you have any questions. Go to quorum sensing now.
Sir, please tell me then how they can pass blood-brain barrier, listeria?
Oh, how do they pass? Boy,
Yes, sir.
it will take an hour to explain you. No, blood-brain barrier also like a gut barrier.
Okay.
They have a similar structure,
Okay.
and so we believe they have also tight junction proteins, or also similar epithelial structure. And I think we are working on it right now. Other people have also worked on it, And so other people showed that internally in ape, one of the Listeria proteins, help bacteria to go through the cross the blood-brain barrier. But in our studies also we know LAP binds to this HSP60 molecule, and also it can activate signaling cascade or myosin light chain kinase, because those proteins are also involved. And so if it opens up, so it will be like a gut and the brain very similar way. So this may be playing important role. Then other thing is that how do they cross also the placental barrier, because for the fetus. And that mechanism would be slightly different because different types of cells are involved.
Okay. As you mentioned, the M cell is the natural phagocyte.
Yeah, phagocyte.
Yes. Then how can they pass Listeria inside the body? Means it's a natural phagocyte. Then its main work to destroy the Listeria. Then
Yeah, yeah. So anytime we have a disease, right? So that means our immune system failed, right? That means immune system could not contain the infection. So the Listeria being intracellular pathogens, when they get into the macrophage and they get out of the vacuole very quickly, and then immediately they start multiplying in the macrophage, and then eventually they kill the macrophage. Okay, if the macrophage is dead, obviously the bacteria is going to survive very well. And so the M cell is a natural phagocytic cell, but the problem with the M cell doesn't kill bacteria. It's kind of like a passageway. Okay, but as soon as they pass, then the phagocytic cells, whether it's a dendritic cells, then the macrophage or neutrophil or other cells, they'll try to contain it. But thing is, because Listeria can survive inside, they can kill the immune cells, and that's why they can persist the infection. So anytime your immune cells fail, that's when you become sick, right? Otherwise, if you are not sick or under, little bit of under the weather, that means your immune system trying to work to contain their infection. But if you have a full-blown infection, means your immune system could not do its job. It could not protect you.
thank you, sir.
Okay. Any other questions? Okay.
Good morning, sir. So
Yeah.
my question is, can sessile phenotypes exist without the extracellular matrix?
Say that again. Can you repeat that?
Can sessile phenotypes exist without the extracellular matrix?
Slow down a little bit. Cell phenotype, serotype.
Sessile phenotypes. Can they exist without the ECM, extracellular matrix?
Which bacteria? I mean, you are talking about Listeria?
Yes, sir.
Oh yeah, they can survive without the extracellular matrix. But you are saying that sessile cells, when we are collecting from the biofilm,
Yes, sir.
yeah, there is, when you isolate them, obviously those extracellular matrix are washed off. So that is the question you are asking, or the
Yes, sir. This one.
yeah. So that's the difference. Yeah, it's not truly we are injecting biofilm, right? What we did, we scraped off, and it become a cell suspension. Okay, and those the cells we are taking it. But without the EPS, they're still maintaining their, that sessile conditions. You see this? So the sessile conditions would be the genes are down-regulated. They are not producing all those virulence factors or proteins. They're still maintaining for certain duration. But you have to be careful how you keep them. You have to maintain in a water or buffer without giving them any nutrient or food, and so that they maintain their sessile status. Yeah,
Thank
they
you,
would
sir.
survive. Okay. Any other questions? So I will ask you a question. So I think he brought up a good point. So if you have a pure water, sterile water, right? So you have a deionized water we use, right? And sometimes we call double deionized water, right? No ionic species, nothing. So if you add bacteria, okay, do you expect bacteria to grow in that water sample? It's not a tricky question. Got a pure deionized water, right? Assuming there is no nutrient, nothing, not even ions, right? Deionized, you remove the ions. So if you have a bacteria, freshly grown bacteria you had, say you added, 100 cells into 1 milliliter of water. And I ask you, tell me if this microbe is growing in that water. Yes or no? Say that again. So you are saying they are not going to grow. Yeah, so they are not going to grow. That's what you are saying. But these are fresh cells. I took it from a broth. Huh? Okay, so you are saying they are going to. Okay, anybody else? That's one your answer. That's right. So what's your answer? Is it going to grow or no growth? No growth. Anybody else? Huh? He's smart. So some of you are going to say yes or no, right? Overall, you are saying no, right? Okay. So here is the other thing you got to think about. So the bacteria you harvested from a fresh media, they grew, right? So they are healthy cells. Now I took these cells and put it in water, right? Deionized water. There is no nutrient, nothing. So a question I was asking, are they going to grow? Right? Some of you say no, because there is no food, right? We will learn that. That's true. There is no food in that water, right? But this bacteria came from healthy environment, right? They are going to multiply at least until they exhaust their stored energy. So the answer is yes, limited growth. They may grow a little bit, then after that they're going to stop because they have the stored energy. So a lot of times I think students do a mistake. It's a perception that, oh, they are not going to grow. But you are forgetting they can multiply their number. So how you design your experiment, even in the buffer they can multiply, they can misrepresent your number. Keep that in mind. All those little tricky things, they can multiply to an, if you have a 10 to the power 6, they can double it. Right? 1 times 10 to the 6, they become 2 times 10 to the 6. So that's a huge difference. 1 million to 2 million cells. Okay. Everybody got it? Those who are that side, I think you see this what I was talking about? The bacteria they need food, right, to grow. But in this case, they already have their stored food. Like we can skip a meal or 2 meals, 3 meals, we can still survive, right? Because our stored energy. And the bacteria, when they're growing in rich media, they harvest out of it, they store some sugar inside. They can still continue their process at least for 1 generation or 2 generations. So they will multiply at least 1 time or 2 times depending on how robust they are. Otherwise, if you have a starved cell or sessile cells, you put it in the water, no, I don't expect them to grow. You see this? So it's all physiology. Okay, I thought I will ask that question. It's not tricky question though. So we're going to talk about the quorum sensing. I know this presentation should not be that long, and I know I'm pretty near, but we'll try to stay on time to give you the break. So quorum sensing and the regulation. So from Monday morning all the way to yesterday evening, everybody talked about quorum sensing, right? It's a language, it's a vocabulary of biofilm, right? Anytime you talk about biofilm, you cannot avoid the word QS or quorum sensing, right? It's the cell-to-cell communication. Everybody tells you that. So we'll learn a little bit today, okay? So part of the biofilm formation is that attaches, and attachment is important, right? We all learn that. It's critical. And then it have a quorum sensing, means it communicate each other. And then we need to understand how the biofilm formation regulated, right? And we'll talk about cyclic di-GMP. So attachment we mentioned repeatedly yesterday, a lot of virulence factor, a lot of proteins, molecules, they are produced by the bacteria, and Carli, fimbriae, flagella, proteins, teichoic acid, I already mentioned that, right? And these help the bacteria to attach to the surface. And so here, before I get into quorum sensing, I want to give you a little bit of background how these proteins are made. Okay, one aspect I kind of introduced protein secretory pathways, right? So I want to ask you, or how do these proteins are produced before they are available for attachment? Okay, and again, these are more advanced knowledge. You need to understand And these are important. These are the way, these are the direction research are going. So this is called protein secretion system. So bacteria or any microorganisms or our cells, they synthesize proteins inside, right? Even the curly or flagella, all of those molecules are synthesized inside, and then they are exported outside the cell. Okay. The question is, how do they get out of the cell? Okay. And there is a special machinery help the bacteria to release these molecules. Okay. Or protein secretion system. So, for example, we went through this gram-positive structure, right? So you have a cell wall, right? That's the peptidoglycan structure. Everybody follow this. Then you got the cytoplasmic membrane, and inside, this is where these proteins are made. So the proteins are inside. So if it is a flagellar protein, for example, right, or it's a fimbrial protein, they are made inside. How do they get out? See this protein, see this is called exoprotein. Have you ever given thought how these proteins are getting out of the cell? Okay, and you've got to have some mechanism how they get out. Okay, so same way in the gram negative. Gram negative even difficult, right? You got extra layer. So you got outer membrane, then the plasmic space, then this is the peptidoglycan layer or cell wall, right? And then the cytoplasm. So look at these proteins are here. How do these proteins get out? There are 3 layers, right? So this is always an important question. What about the mycobacterium, right? Mycobacterium has again cell wall, but it has this lipid layer outside, extra layer, right? And mycolic acid layer, and then the cytoplasmic layer. So how do these proteins get out of this cell? So always a huge question. And then if these proteins are not outside, they cannot use them for pathogenesis. Are you following me? Those are important virulence factor, right? So people study this, how these proteins get out, for the purpose of, if you want to develop a vaccine or if you want to develop a drug, can I block this protein export? So think this number of ways. Everything we are learning try to prevent infection, prevent disease. All your goal would be also, right, I'm trying to study these bacteria or how these proteins are made, because eventually I want to make a vaccine or I want to develop a drug to prevent this protein from getting out. If they stop outside, then they will be less invasive or less infective, or they cannot form biofilm. Are you seeing the picture now? Okay, so that was the reason we want to know this. So I know this picture is a little bit colorful, but complex, right? So we call it protein secretion system, right? I already mentioned that. So there are different types of protein secretion system. So there is a T1 through T9. So type 1 secretion system called T1SS. Okay, and if you look at this in the gram negative, that's where the most complex cell architecture, right? Because you got outer membrane, then you got the cytoplasmic membrane, and then the cell wall in the middle, right? So the question is, how do this protein get out? And look at this, all those proteins. Adhesin, we mentioned about BAP yesterday, right? Few times, bacterial or biofilm associated protein, pili, flagellum, adhesins, all of those, curly, all of those molecules, how do they get out? So a lot of you, I think I talked to you, you guys said, well, I want to do research, how do I block or I go to prevent biofilm formation? Well, I'm going to work on, say, plant extract, for example, right? A lot of times people are looking for bioactive molecules, and if I add that, can it prevent biofilm formation, right? So number of ways you could apply. Look at these drugs or the molecules, can it prevent their secretion of these molecules which are essential for attachment? And if you prevent attachment, of course you are going to prevent their biofilm formation ability. Are you seeing this? This is how you have to connect it. Okay? And so here I want to show you this type 1 through type 9. So this is a type 1. It's almost like called ABC transporter, and it's like a channel. Okay, so proteins are made, it goes through this and get out. Okay, and then display on the surface. Then type 2 also, and that is called sex system, type 2 secretion system, type 3 and type 3b, type 4 and type 5, type 6, type 7, type 8. So essentially all of them maintain some kind of general structure. It's like a create a tube, right? If you are trying to pass water or air from one room to other room, you use a tube, right? So bacteria, they are using some kind of channel, and that's how they drive their molecules. Okay. So the type 1 and type 2, a lot of this kind of releasing outside. One of the most important one we discussed quite a bit in the disease mechanism is called type 3 secretion system. This is the key, type 3 for many diseases. What it does actually, type 3 secretion system, you heard about the syringe and needle. When you go to see a doctor, what they do? They inject you medicine, right? How do they do that? Use a syringe, right? And so type 3 secretion system is like bacteria use this syringe, directly deliver its virulence factor inside the cell. Okay? Directly. The reason is they don't want to get lost. If it is not directly injected, it can get lost. Okay? So I think that's the one. And so if this is flagellar synthesis, and this is the called type 3 secretion system, which directly inject the proteins into the host cell. And I think these are some actually well studied. These are actual electron microscopy photograph of type 3 secretion system in bacteria. These are, I think, could be from Salmonella. A lot of these molecules are produced by Salmonella and directly inject into the epithelial cell. This is how they look like. The cytoplasmic membrane, cell wall and inner structure, outer membrane, and then it injects directly. This is actually model it, you look like this. Okay? So the whole idea being is that gram-negative organism, gram-negative, all the bacteria, notice the Salmonella, E. coli, Escherichia, Campylobacter, these are Vibrio, these are all gram-negative organisms. They have 3 layers, and they use this type 3 secretion system to inject a lot of these molecules. But the reason I am bringing this up for biofilm formation, I already mentioned that these molecules can help release a lot of these molecules outside, okay, for purpose of using them as adhesion factor. I know we need to take a break. It's 11 o'clock. Let's take a break, then we'll come back, then talk about from here, and we'll finish that. Okay? Okay? Take a break, and then during break or after break, you can ask me questions. I'm available. Or if you are here, we'll talk about it.